Hi guys, welcome back to my channel. This is Dr. Rajeshwar from YR PharmaTube. In the previous video, we have learnt the phase 2 sulfate conjugation reaction. If you did not watch the video, hit the I button on top right of this video to watch it. To watch the other topics of drug metabolism and the other subjects of pharmaceutical sciences, click on the links given in the description box below this video. In this lesson, we shall try to understand the phase 2 methyl conjugation reaction of drug metabolism. At the end of this video, you will be able to understand the methylation conjugation reaction of phase 2 drug metabolism. Methylation is a chemical reaction in which a methyl group is added to a substrate or a drug molecule, or the substitution of an atom or group by a methyl group. Methylation is a form of alkylation with a methyl group replacing a hydrogen atom. Methylation is extremely important in the biosynthesis of nucleic acids, proteins, phospholipids, neurotransmitters, and other amines. In contrast, methylation is a minor metabolic pathway that is seen in the xenobiotics or the foreign compounds that contain catechol, phenoxyhydroxyl groups, amines, also N-heterocyclic compounds, and sulfhydryl groups. Methylation reactions onto nitrogen, oxygen, and sulfur functional groups all utilize S-adenosyl methionine as the methyl donor, but they are catalyzed by different enzymes in each case. Methylation reactions are involved in the biosynthesis of many endogenous compounds like epinephrine and melatonin, and in the inactivation of numerous physiologically active biogenic amines such as norepinephrine, dopamine, serotonin, and histamine. These reactions constitute only a minor pathway for conjugating drugs and xenobiotics. In general, unlike other conjugation reactions, methylation does not lead to polar or water-soluble metabolites and thus hinders excretion of the drug, except that when methylation creates a quaternary ammonium derivative. Most methylated products tend to be pharmacologically inactive, although there are a few exceptions. Mechanism of methylation. The methylation of functional groups occurs via in a two-step process as shown in the figure. Methyl group is provided by the amino acid methionine and it must be activated to transfer its methyl group. The enzyme methionine adenosyl transferase uses methionine and ATP to synthesize S-adenosyl methionine, commonly abbreviated as SAM. SAM can also be considered as a high-energy intermediate similar to UDP glucuronic acid and PAPS as discussed in the glucuronidation reaction in the previous video. The positively charged sulfur atom is electrophilic in nature and allows for the easy transfer of the methyl group to a catechol, a phenolic hydroxyl group, an amine, or a sulfhydryl group. Then S-adenosyl homocysteine can be converted back to methionine in two steps to allow additional methylations to occur. A list of methyl transferases and their substrates is given in the table. These transferases transfer methyl group from coenzyme S-adenosyl methionine to the drug molecules or their metabolites. Methylation reactions. There are three types of methylation reactions, namely O-methylation, N-methylation, and S-methylation. Methylation reactions onto nitrogen, oxygen, and sulfur functional groups all utilize adenosyl methionine as the methyl donor, but they are catalyzed by different enzymes in each case. O-methylation. O-methylation is a common reaction of compounds containing a catechol ring, that is, an aromatic ring with orthohydroxyl groups. These compounds are most likely to undergo phase 2 conjugation reactions. This includes endogenous amines such as epinephrine, norepinephrine, and dopamine, as well as a number of adrenergic agonists such as methyldopa and isoproterenol. Catechol O-methyl transferase, abbreviated COMT, a magnesium-dependent enzyme, transfers methyl groups specifically to the meta position of the catechol derivatives such as epinephrine and isoproterenol. Hydroxyindole O-methyl transferase catalyzes methylation on N-acetyl serotonin to melatonin, a pineal gland hormone. Phenylethanol N-methyl transferase is the enzyme responsible for catalyzing the biotransformation of norepinephrine to epinephrine. It will similarly methylate synthetic phenylethanolamine drugs such as the decongestant agent phenylephrine. However, more frequently, O-methylation occurs as a late event in the metabolism of aryl groups after they have been oxidized to catechols. This sequence was seen, for example, in the metabolism of anti-inflammatory drug diclofenac, which in humans yielded 3-hydroxy 4-methoxydiclofenac as a major metabolite with a very long plasma half-life. The methylation of catechol rings serves a second purpose because it prevents their oxidation to highly reactive intermediates as shown here. As previously discussed, glutathione can help neutralize or inactivate these types of reactive intermediates. However, without COMT, high doses of catechol containing drugs could deplete glutathione stores. Please note that the methylation of catechols occurs preferentially at the meta position and that the N-methylation of phenylpropanolamine is a minor metabolic pathway for this drug molecule. Due to the availability of other metabolic pathways, phenolic hydroxyl groups are only methylated to a minor extent. One interesting example involves the conversion of morphine to its O-methylated derivative codeine in humans. This metabolite is formed in significant amounts in tolerant subjects and may account for up to 10% of the morphine dose. N-methylation. The methylation of amines is essential for biosynthetic pathways but of very limited importance as it relates to drug metabolism. As previously discussed, amines are primarily dealkylated and are deaminated. Three basic types of N-methylation reactions have been recognized and are shown in the figures here. These reactions are catalyzed by N-methyl transferase enzymes. Specific N-methyl transferase enzymes catalyze the transfer of active methyl groups from S-adenosyl methionine to the acceptor substance. The non-specific N-methyl transferase found in the lung can reverse the N-demethylation reactions of phase 1 metabolism. But most of the other methyl transferases are specific for endogenous compounds except the S-methyl transferase that is found in the microsomal fraction and which will methylate many thiols such as thiouracil. N-methylation in drugs containing primary and secondary amines. Drugs or xenobiotics containing primary and secondary amines, that is, some phenylethanolamines and tetrahydroisoquinolines, are the substrates of N-methylation reaction. These compounds undergo N-methylation using N-methyl transferase enzyme. Although N-methylation of endogenous amines such as histamine, norepinephrine, dopamine, serotonin, tryptamine, tyramine, etc., and drugs like amphetamine, normorphine, desmethylimipramine, etc., occurs commonly, biotransformation of nitrogen-containing xenobiotics to N-methylated metabolites occurs to only a limited extent. Thus, these reactions have rare significance in vivo, presumably due to effective oxidative N-demethylation. It seems that Amine N-methyl transferases have a role in recycling N-demethylated drugs. For example, histamine N-methyl transferase methylates specifically histamine, producing the inactive metabolite N-methylhistamine. In the other example, noradrenaline is converted into adrenaline by the action of methyl transferase enzyme. Phenylethanolamine N-methyl transferase methylates a number of endogenous and exogenous phenylethanolamines such as normetanephrine, norepinephrine, and norepinephrine, but does not methylate phenylethylamines. The other example is the antiviral and anti-Parkinsonism agent amantadine in dogs and the in vitro N-methylation of norepinephrine in rabbit lung preparations. N-methylation in imidazoles. The NH group in an imidazole ring can also undergo N-methylation. For example, histamine can be converted to its N-methyl conjugate by N-methylation reaction. In another example, theophylline, whose N9 methylation is masked by N-demethylation in adult but not newborn humans. N-methylation in pyridines. N-methylation of nitrogen atoms present in heterocyclic compounds, that is pyridine derivatives, also takes place. N-methylation of pyridine-type nitrogen atoms appears to be of greater in vivo pharmacological significance than the reactions discussed above, and this for two reasons. First, the resulting metabolites, being quaternary amines, are more stable than tertiary and secondary amines toward N-demethylation. And second, these metabolites are also more polar than the parent compounds in contrast to the products of N-methylation in primary and secondary amines and in imidazoles. Good substrates are nicotinamide, pyridine, and a number of monocyclic and bicyclic derivatives. For example, the pyridinyl nitrogens of nicotine and nicotinic acid are N-methylated to yield quaternary ammonium products. S-methylation. Compounds containing thiol or sulfhydryl groups also undergo methylation to form S-methyl conjugates. These reactions are catalyzed by S-methyl transferase enzyme and also involve a microsomal enzyme requiring S-adenosyl methionine as discussed in the mechanism of methylation. S-methylation of thiol groups is documented for such drugs as captopril and 6-mercaptopurine. In addition to 6-mercaptopurine, thiopurine methyltransferase or thiopurine S-methyl transferase, that is abbreviated as TPMT, also catalyzes the S-methylation of thiopurine drugs such as azathioprine and 6-thioguanine. Inter-individual variations in sensitivity and toxicity to thiopurine are correlated with TPMT genetic polymorphisms. Defects in the TPMT gene lead to decreased methylation and decreased inactivation of the thiopurine drugs, leading to enhanced bone marrow toxicity that may cause myelosuppression, anemia, bleeding tendency, leukopenia, and infection. Approximately 5% of all thiopurine therapies will fail due to toxicity as a result of TPMT polymorphism. Thiols are toxic, and the role of thio-S-methyl transferases is a monooxidative detoxification pathway of these compounds. Although a wide range of exogenous sulfhydryl compounds are S-methylated by this microsomal enzyme, none of the endogenous sulfhydryl compounds, for example cysteine and glutathione, can function as substrates. Clearly, S-methylation represents a detoxification step for thiols. Other substrates are metabolites, mainly thiophenols, resulting from the sulfur-carbon cleavage of aromatic glutathione and cysteine conjugates. Once formed, such methyl metabolites can be further processed to sulfoxides and sulfones before excretion. From the reaction as given here, it is apparent that methylation reactions can be subdivided into two classes. Number 1, those where the substrate and the product have the same electrical state, a proton in the substrate having been exchanged for a positively charged methyl group. And number 2, those where the product has acquired a positive charge, namely has become a pyridine type quaternary ammonium ion. Dialkyl dithiocarbamates, for example disulfiram, and the antithyroid drugs, for example propylthiouracil, mercaptans such as dimercaprol and hydrogen sulfide from thioglycosides as natural constituents of foods, mineral sulfides in water, fermented beverages, and bacterial digestion are all S-methylated. Due to their low prevalence in drug molecules, S-methylation is observed rarely. Methylation does not increase hydrophilicity because methyl group is lipophilic in nature. Most methylated products tend to be pharmacologically inactive with a few exceptions. Methylation can be reversed through the process of oxidative dealkylation. However, this does not occur very often. In general, unlike other conjugation reactions, methylation leads to a less polar product and thus hinders excretion of the drug. This is the list of references followed for the lecture. That's all in this video. The methyl conjugation, a type of phase 2 drug metabolic reaction. In the next lesson, we will discuss about the phase 2 acetylation reaction. Till then, never stop learning and never stop watching my videos. Thank you for watching this video.